Hello everyone, in this video I will explain cholesterol biosynthesis. Cholesterol is a sterol exclusively found in animals. It can be taken in the diet but it also can be synthesized by the body. All tissues are capable of synthesizing cholesterol, but largest contribution is made by liver, intestine, skin, adrenal cortex, reproductive tissue, etc. Cholesterol in the body is transported in the form of cholesterol ester. That is 70% of the cholesterol is transported in the esterified form, that is cholesterol ester. How this cholesterol ester is formed is when cholesterol combines with phospholipid, that is lecithin, it forms its transportable form, that is cholesterol ester and also lysolecithin. Only 30% of the cholesterol is transported in a free form. Coming to the biosynthesis of cholesterol, it takes place in cytosol. That is, the enzymes that are required for cholesterol biosynthesis are present in cytosolic fraction of the cell. Now we will see how this cholesterol synthesis takes place and what are the reactions that takes place during biosynthesis of cholesterol. Acetyl-CoA is the precursor for cholesterol biosynthesis, and we know that how the body gets this acetyl-CoA. During glycolysis, pyruvate is formed. That pyruvate gets converted to acetyl-CoA, and not only that, certain amino acid degradation will yield acetyl-CoA, and also oxidation of fatty acids will also give acetyl CoA. So this acetyl CoA is the precursor for cholesterol synthesis. And this acetyl CoA is a 2 carbon compound. From this 2 carbon compound, 27 carbon cholesterol is going to be formed. And coming to the reactions, the very first reaction is two molecules of acetyl CoA combines in the presence of thiolase enzyme and forms acetoacetyl-CoA. This acetoacetyl-CoA condenses with one more molecule of acetyl-CoA and forms HMG-CoA. This reaction is catalyzed by HMG-CoA synthase. And HMG-CoA is beta-hydroxy beta-methyl glutaryl CoA. And next reaction is a reduction reaction. HMG-CoA undergoes a reduction to mevalonate in the presence of an enzyme HMG-CoA reductase. And this reaction is a rate limiting step in cholesterol biosynthesis, and HMG-CoA reductase is rate limiting enzyme in cholesterol biosynthesis. And it is a very good example for a simple feedback mechanism. That is, whenever the concentration of cholesterol is increased, it starts inhibiting this enzyme HMG-CoA reductase starts inhibiting its own production, cholesterol production. That is meant by feedback inhibition. And also the statins are competitive inhibitors for this HMG-CoA reductase. So that is the rate limiting step of the cholesterol biosynthesis. And next reaction is this mevalonate undergoes 3 phosphorylation reactions and forms isopentenyl pyrophosphate. This isopentenyl pyrophosphate isomerizes to dimethyl allyl pyrophosphate. These two are 5 carbon compounds, and then these 5 carbon compounds, isopentenyl pyrophosphate and dimethyl allyl pyrophosphate, forms geranyl pyrophosphate, which is a 10 carbon compound. And next reaction of the cholesterol biosynthesis is geranyl pyrophosphate combines with isopentenyl pyrophosphate molecule and forms farnesyl pyrophosphate. This takes place in the presence of transferase enzyme. Farnesyl pyrophosphate is a 15 carbon compound. And next reaction is two molecules of farnesyl pyrophosphate condenses in the presence of squalene synthase and forms squalene, which is a 30 carbon compound. And this next reaction is squalene undergoes uh, reaction and forms lanosterol, which is a 30 carbon compound. This lanosterol undergoes 19 enzymatic reactions and forms cholesterol, which is a 27 carbon compound. So, this is about cholesterol biosynthesis, which is having three 6 carbon rings and then one carbon ring, one 5 carbon ring, and then which is followed by the carbon chain, which is of total, which is consisting of total 27 carbons. So if you see here, so 1, 2, 3, 4, 5, then 6, 7, 8, 9, 10, then 11, 12, 13, 14, then 15, 16, 17, 18, then 19. This one is 20, 21, 22, 23, 24, 25, 26, and 27. In that way, 27 carbon compound, that is cholesterol, is synthesized from the 2 carbon acetyl CoA molecule. Coming to the functions of the cholesterol, so this is a structural component of the cell membrane and precursor for the synthesis of all other steroids in the body. These include steroid hormones like aldosterone, cortisols, progesterone, estrogen, testosterone, etc. And also it is the precursor for vitamin D and bile acids. And this is an essential ingredient in the structure of lipoproteins, in which form the lipids in the body are transported, that is cholesterol ester, and also the fatty acids are transported to liver as cholesterol esters for oxidation. So this is about this video is regarding the biosynthesis of cholesterol and the functions of cholesterol. Thank you.